and we found that about 50% of them have encountered mutation in this highly conserved region. So, but the take-home lesson from this particular mutation is that the triple G is very important and significant for the viral replication. So from this point we started off trying to design an antisense drug or antisense molecules and to test whether these antisense could inhibit viral replication in vitro. So we decided to design two antisense molecules. One of them is our control that was designed against the initiating methionine, and the other one was against the 3D structure, including the very important phylogenetically conserved triple G. This cartoon will show, I think Dr. Stuyton showed us how these antisense molecules work. It blocks or it interferes with the translation machinery so that the RNA cannot be translated, and also it triggers the intracellular RNase H that degrades the RNA involved in the heterodimer between the short DNA molecule and the RNA of the virus. So we wanted to establish an in vitro system to propagate HCV to study the effect of these antisense molecules. So most of the people use the transfection experiments in which they use either full-length HCV cDNA or they in vitro transcribe this cDNA into a viral RNA, or they use subgenomic HCV fragments and they link these to a reporter gene with a luciferase activity that can monitor the infectivity of the replicon into the cells. However, we found that this strategy has some disadvantages. First, it is not permissible to the viral replication, and because it starts with a cDNA, it lacks both the 5' and the 3' non-coding regions, which is the focus of our study, and also due to the lack of fidelity of the RNA-dependent RNA polymerase of the virus, it incorporates lots of mutations with a chance to incorporate artificial splicing sites, so the RNA is sensitive to intracellular spliceosomes and the processing, so degradation is also possible for this molecule. So we decided to go back to the natural way of infecting the monolayer cells with HCV positive sera, and we had options to use hepatoma cells or hepatoblastoma cells or fetal hepatocytes or fused primary human hepatocytes with hepatoblastoma cells. We decided to use the HG2 cells because that they have more than one advantage. They are the closest of the cell lines to the normal hepatocytes. They have the LDL and the CD81 receptors, which many investigators reported that these receptors mediate the entrance of HCV particles into the cells. Most importantly, it has been reported from several labs the presence of a 66 kilodalton membrane protein on the membrane of these cells that act as a receptor for the antisense molecules. So it is good for our particular purpose. These are the HG2 cells, and these are the antisense that we designed by the board. This is the SODN2 and this is the SODN1. This includes at the beginning of the core gene, so this is the initiating methionine, and this is the triple G at the apical part. It is highly conserved among all the quasi-species that we analyzed from our local strains. So after infecting the cells with the antisense, with the virus, in presence and the absence of the antisense molecules, and following the cells for 48 and 72 hours, the first antisense compound against the AUG initiation codon at 2 concentrations, 1 micromolar and 2 micromolar concentrations, could inhibit both the synthesis of plus and the minus strand. Actually, the virus, hepatitis C virus, replicates through an RNA intermediate known as the minus strand. So the presence of the minus strand indicates that the virus is replicating. So both the concentrations used, 1 and 2 micromolar, could inhibit completely the synthesis of both plus and minus strand after 48 hours and 72 hours. The other antisense that we developed against the stem loop structure was not as efficient as the one that was developed against the cis-acting element. However, after 72 hours, both concentrations could inhibit the first antisense molecule. So both antisense molecules at the two used concentrations were effective against the HCV replication. And this is real-time PCR demonstration of the intracellular quantitation of the virus to show that our RNA is intact among all the treatments used, and also that to show that the antisense molecules was selectively reacting with the HCV sequences. We used housekeeping GAPDH, glyceraldehyde 3-phosphate dehydrogenase, to check the mRNA levels for this housekeeping gene, and it was actually fairly fluctuating over all the treatments using either concentration of the two antisense molecules. It was ranging from 100 to 180 nanograms per ml in all the treatments, which means that our RNA preparations were intact, and the activity of the antisense molecules was selective towards the HCV sequences. Of course, there are lots of experiments that we are involved in right now to check whether these will affect cell cycle regulating genes and probably other apoptotic genes. So we will do lots of control experiments to check the effect of these antisense molecules. At this point, we do not know what to do with this. Hopefully we will start moving toward trying to develop delivery systems or trying to collaborate with someone to use the most optimum delivery system for these and move a little bit from the in vitro situation to chimpanzee, for example, which is the only non-human host for the HCV. Some pharmacokinetic studies that these SODNs, when injected into the small experimental animal, within 2 hours, 20% go to the liver, which is our target tissue, our preference target tissue, and 80% will go to the other tissues. 50% will be degraded in 2 days. As far as elimination of these compounds, in the first 24 hours, 30% will be eliminated, 70% will take 10 days to be eliminated. Tissue distribution of the compound will was fairly equal between the different tissues. And this is good because hepatitis C virus is not only hepatochronic virus, it infects also the bone marrow and probably other tissues. Kidney also incorporates HCV. Skin may have HCV. Some other tissues. Brain has little amount of the injected SODNs due to the blood-brain barriers or whatever. But probably during the delivery systems we can use targeting molecules toward preference tissues like liver or any other tissues. But the native SODN can be distributed fairly between the different tissues. So in conclusion, the translation of HCV proteins is regulated by both cis and trans-acting mechanisms via the 5' UTR region. Also, the natural mutations that we have seen in the healthy carrier patients within the 5' UTR surely interfere with the life cycle of the virus via inhibition of efficient translation of HCV polyprotein precursor, and eventually they will affect the synthesis or the expression of the structural as well as the non-structural proteins. Therefore, the designing of antisense SODN compounds against the 3D stem loop and the AUG start codon may be efficient in HCV control in vitro and probably later on in vivo. Thank you very much.
Thank you very much, Dr. Mustafa. We have the first question from a clinician.
Okay.
How do you think tissue engineering served in such a big problem in Egypt?
I just told you a few before I get